Most students find anatomy to be pretty straightforward. There's a lot of memorization and there's some thinking involved, but nothing like what's ahead of you in physiology. And one of the most challenging things about physiology is that you can't see it. You can't see molecules. You can't see what's going on in a cell, even though you can see, as you learn a little histology, you can learn about cells. So what I want to walk you through is some of the ways we have for visualizing molecules. There are two types of models that I've found for you that represent some of the most common molecules and small ions that you'll interact with throughout the course of the semester. I have actually only one or two of these. These are ball and stick models, and each of the balls in this particular model represents an atom, and then the sticks represent bonds. So there's a bond there, and there's a bond right there, okay? So the more common models that I'm going to show you today are the ones that are called space-filling models, where instead of trying to show the covalent bonds that hold all these molecules together, they've settled for showing you what the overall model looks like. I know you know that one because you've seen it enough. Red is typically oxygen, and white is typically hydrogen. This one's a freaky example over here that doesn't use the same color coding system that we typically use. Often black is for carbon, blue represents nitrogen, yellow or an orange is phosphorus, red is oxygen, and white is hydrogen. That's kind of the theme that most of the models follow. So we're going to sort of take a tour of common molecules that we'll see throughout the course of the semester. And in the course of this tour, I'm going to try to tell you a story that I hope will help it all hang together for you. This is biology, of course, so all of the chemistry that we're talking about today is set in this context. And there are some of the features of this cell that are really, really important to us, like the plasma membrane right here, the cell membrane. Here's a mitochondria, number 9. You should know all of these, but we're going to focus in on the ones that are right here. And I'm saying this because this is the context of the story that I'm going to tell you, onto which I hope to hang all of the molecules I want you to learn a bit about. Take a moment to look at the features of the cell that I've kept on my model. There's a vesicle, mitochondria. I don't really need a vacuole. The cytosol, that's the cytosol, and here's the cell membrane. So what I want to do is introduce you to some of the different classes of biological molecules, and I want to do it by looking at the function of the cell. So you know, for example, that the mitochondria, it's all about energy. Where do we get the energy? Well, we get it from decomposition of molecules like glucose. We get the glucose into the cell. It goes through a metabolic pathway called glycolysis. Do you remember that? And then the remnants of that glucose molecule enter the mitochondria, and they go through a really elaborate metabolic pathway that's called oxidative phosphorylation. It is at the cellular level, the reason we need oxygen. It's not the only use of oxygen in our body, but it's a huge one. And the CO2 that we have to exhale, that's going to come from the final remnants of the glucose molecule, the carbon backbone of that molecule. And why do we do all of this? Because we're trying to charge the battery here. I like to call ADP the uncharged battery, and this arrow here represents the addition of energy and the enzymatic conversion of ADP to the fully charged version of this battery, which is ATP. So that's part of the story. The other part of the story is that if you are particularly well fed, like right after you've had a meal, you don't actually need all of the glucose to go through the metabolic pathways that lead to production of ATP. And in fact, two organs in particular, the liver and to a lesser extent skeletal muscle, are going to, I like to say, squirrel away that glucose. They're going to convert glucose, which is a monomer, that's monomer. That's ugly, but it looks like the word, to glycogen, which is a huge polysaccharide. Again, one of the things I'm trying to illustrate is the difference in size of all of these molecules. I will never illustrate my ability to spell. You're just going to have to deal with it. I went out onto the web for you and spent a stupid amount of time looking for space-filling molecules for particular things that I was interested in. And in order to try to put these at roughly to scale, I just chose the size of the circle and said, okay, I'm going to see if I can make this circle represent the size of an oxygen atom. And in all of these molecules, I tried to get the oxygen atoms to roughly that scale. It's not a beautiful system, but at least it roughly shows you that among the things we talked about on that previous slide, and water is outside that cell everywhere, and it's inside, it's in the cytosol of that cell, so I thought I'd include it here. Water, oxygen, CO2, this is C and O, right? And glucose. Glucose is a very large molecule, isn't it? It has a number of black, see the carbon atoms down in there? And a lot of these OH, right? That's O and that's H groups sticking out of this carbon ring. This is ATP. Do you remember ATP is, I called it a nucleotide over here. It's really not quite a nucleotide, but remember it has a nitrogenous base. So those N, the blue ones are N. Here's carbon. Each of these groups here is what's called a phosphate group. So the yellow is P surrounded by oxygen. This has 1, 2, 3 phosphate groups, like so. So that's ATP. Okay, now we're going to try to see what it takes to convert glucose to glycogen. Now, what typically happens, and I say typically happens, it always happens for glucose to glycogen, but in all of our classes of biological molecules, there is a monomer, and in this case, that monomer is a monosaccharide, and you add one glucose molecule to another glucose molecule, and you would get a disaccharide. If you keep going, if you add many glucose molecules, you will eventually get glycogen, which, as I said before, is a polysaccharide. I'm actually going to write it somewhat readably here. How large is glycogen? It's so large it won't fit on my slide. This is part of a sphere, so it goes all the way around, right, way off the slide. And it's a cross section, which means, take a gumball or whatever and slice through it, and actually slice through it again, so you get one thin slice, like deli slice, and this is what you see, a layer of a spherical construct. This is glucose, and this is roughly a glucose molecule. Here's another one. Here's another one. You see what this is? So it's extraordinarily large. It has here's a long chain of glucose molecules branching off of another chain of glucose molecules. And in the middle of this sphere, these long chains of sugars, of glucose, are attached to this sort of core proteins in the middle that organize the whole thing. You don't need to know this, but that's this protein that is making all of this happen. And in fact, this glycogen granule would come together with other glycogen granules, and they're so large that, well, let me show you how large they are. This is a liver cell, or a hepatocyte, that is magnified 5,000 times. To do that, you need an electron micrograph and an electron microscope. And let's see if you see what I see. This thing right here, this is the membrane, the very edge of the nucleus. And all of these guys, this one here, these big dark things, even though they don't all look the same, sort of looks like an astronaut's footprint on the moon, that's mitochondria. MT for mitochondria. This scientist who wrote this is using the term GER, granular endoplasmic reticulum, to name something that we call rough endoplasmic reticulum. So it's endoplasmic reticulum and it's got some ribosomes on it. But you see all this over here? All these little black dots here? That is glycogen. So they're granules of glycogen that are so large that they're visible. They look the same size as a membrane. They're huge arrays of glycogen inside the cytoplasm of this cell.